So I'm going to keep this short and just give you a preview of what to expect today, what we, very short recap of yesterday, and some of the exciting things that we have coming up through the rest of the year for us. Okay. So, very quick announcement. As you know, we have a gala later today. We also have a silent auction that we are running. There are several auction items on the table at the right to the back of the room. You can also scan this QR code and look through the website, and there's several interesting items on there. And I'd love it if you could all take a look and give us some love, keep the momentum going. We're very excited about this, and we'd love to have everyone participate and make sure you don't miss out on some of the wonderful items that are there in the auction. And of course, please join us for the gala later today. It promises to be a lot of fun. So very quickly, yesterday and from the day before, we learned several different topics relating to patient-centered outcomes, the entire Parents 2.0 effort. You have heard a lot from the parents who are providing their own stories as part of the symposium as well. We, of course, kicked off the day with Dr. Longo's talk and an overview of CCDS diagnosis tools, and then segued into models relating to creatine deficiencies, different therapeutic approaches. And then we had a wonderful plenary and keynote talk yesterday, followed by the poster session. All this to say that while yesterday was packed and had a lot of topics of interest, we have more of that coming today. We spotlit some of our research fellows earlier yesterday. We have 2 more that we'll be hearing from today. So stay tuned for those talks. For anyone who's attending this symposium for the first time, I do want to mention that we have run an ACD fellowship program for several years in a row. As part of this fellowship, we fund early investigators in the CCDS space working on basic science as well as translational and clinical focused problems. So as you go through these talks, if you are inspired, I encourage you to reach out to us to learn more about this program and what the application cycles for these are. Last year, we also announced our most ambitious fundraising plan yet, which is Race for a Cure. These are our 2026 awardees. You heard from 2 of them yesterday. You're going to hear from 1 more individual today. We are very excited to have been able to fund 2 CTD programs and 1 GAMT program. It was a fairly competitive cycle for us. And so this is our first cohort of Race for a Cure awardees. The goal very much is to keep this momentum going. These are larger multi-year programs, if you will, and our hope is to build on the most promising and innovating research proposals, moving them towards cures, towards clinics. Thank you. So what's ahead? Day 2 talks, we are going to kick off with cognitive outcomes in clinical trials. We have another session on therapeutic development for CCDS. I'm very excited that this year we'll be able to talk through 2 novel approaches for GAMT. And of course, we covered several for CTD yesterday. In addition to that, we'll talk through some of the more recent developments on our registry, case studies, and close out with some clinical focused talks. We have Cerebrain Therapeutics, which we're very excited to spotlight today. They are working on a clinical trial for CTD already initiated in Europe, so we'll hear from them. We'll hear our capstone talk from Judy on the Vigilance study, and then close out with a panel later today. So I encourage everyone to stay through the rest of the program so we have good engagement, and all of your questions and feedback are addressed throughout the day. So I do want to mention in terms of what is to come. There is today, and we are fortunate to have gathered here today to go through all of this exciting stuff. But there's more work always to be done. We at ACD want to keep the momentum going. We announced our ambitious plans last year. You can continue to participate in it. You heard an exciting announcement yesterday as well. These are all representative of all the different fronts that ACD is working on to move forward towards therapies for all of the disorders that are represented by us. So we've committed 1.1 million to the Race for a Cure awardees. We want to continue this effort over multiple years. So if you would like to participate in this, please go and look at our webpage. If you would like more information, please reach out to me or Heidi at any point in time. We'd love to bring you up to speed on all of our efforts. So donate, share, spread the word. Every action gets us closer to our goals. Also, wanted to make a quick plug. This year, we are doing our Walk for Strength in July. So for those of you who would like to participate or are hearing about this for the first time, I encourage you to go to the website and take a look at the campaign pages. We have some shirts already at the back at the store, more to come on that. If you want to learn how you can participate in a walk or set up a new walk in the area that you're from, again, reach out to us, go up to the webpage, and we can provide more information. Our goal, this is also, apart from Holiday Heroes, this is one of our other fundraising platforms. It's our mid-year effort, and it's a great way to get community engagement. So I think that's mostly what I had for today. So I am ahead of schedule, which is good. 5 more minutes saved in the morning. So I think with that, I will kick us off for today by handing the baton to Ashlea. Ashlea is part of our Parents 2.0 cohort. Ashlea is a part of a family of 5 from Kansas. Their youngest son, Eddie, was diagnosed with CTD at 18 months of age. They have 2 older brothers and a sister. The Pfannenstiel family has been navigating life with CTD since Eddie's diagnosis and is passionate about raising awareness as well as fundraising for research in rare diseases. Ashlea, we are so grateful to you for sharing your story with us today. Over to you.
Are we awake yet today? No? All right, so everybody stand up. We're gonna stand up and we're gonna stretch. We're gonna put our hands in the air. And then you're gonna reach into your pockets and you're gonna take out your wallets and donate to CCDS. All right, you can sit. This is Eddie. He is our 6.5-year-old. He's adorable. He was diagnosed at 18 months old after going through genetic testing for epilepsy disorders because his neurologist at the time didn't know what was going on with him. And after waiting weeks after having his first grand mal seizure, he was diagnosed. And we've been on this journey ever since with him. He has 3 older siblings, which are all here today. They're at kids camp. He has a 16-year-old brother, Landon, a 9-year-old brother, Jordan, and a 14-year-old sister, Avery. And he's the baby of the family, so we all love him dearly. Today, Eddie really struggles a lot with communication. He's mostly nonverbal, even though he likes to yell at me all the time, and you'll hear him say, "Mom," a lot these days, or telling me he wants to go outside to play because that's his favorite place in the world. He hates being in closed doors. He likes to be running around outside free. He struggles with expressing his wants and needs and understanding others. He has been using his AAC device now for about a year, which has really helped his vocabulary. We've seen a big boom in his language the past 6 months or so. So he knows a lot. He's showing us he knows a lot. He just can't get those words out. So that leads to a lot of frustration on his part because he just wants to tell us what he wants and he can't verbally do that. He does, though, have a very strong sense of mapping memory. He knows where everything is located the first time we go there. So anytime we're on a car ride, it ends in a lot of frustrated tears because we're not going to go get cookies and McDonald's every day. Usually that ends in we'll kick, we'll scream because we aren't doing what he wants to do. Wants to be independent and be the boss. So it's a daily struggle to get him to understand that not everything is, what he wants to do all the time. He gets frustrated easy, can't wait long. Transitions are really difficult from preferred activities. So if he's really enjoying what he's doing, we can't get him to come off of what he's doing. So lots of tears involved. He usually takes the frustration out on our doors at home. So we like to hit and kick him a lot, which we've been trying to work through those struggles a lot. We try to help him with those emotions, and our kids are such troopers with it. And we know it's hard for them to deal with it, even though they've become really good at tuning him out sometimes. And we were watching The Life of Lucas yesterday, and I look over and my daughter's in tears because she just loves her baby brother so much and wants him to be successful in life. His moods affect where we go, how long we stay somewhere, how our family experiences outings, because when Eddie's done, it means everybody's done. We don't get to stay and hang out as long because he ends up screaming or crying and wants to run away. So we tried going mini golfing, and he's like, I don't like mini golfing, we're leaving. So that usually means our big kids miss out on a lot of opportunities because we really had to structure our day around if Eddie's going to be in the mood to be there or not. And he likes to elope and not stay near us and likes to run away, which we watch doors a lot because if the door is open, he is gone out the door. And we still struggle with daily life skills like bathing, diaper changes, brushing his teeth, washing his hair. He doesn't like doing any of those things. So even bath time, it's just lots of tears involved. He doesn't like it. But I want you today to understand something very important is that Eddie has so much potential, and he shows us every single day that he is capable of learning because he is learning. It's very slow, but we know that he's doing it. It's in his brain. So this is why your work matters, because the work you do every day, it's not just important, it's life-changing to our children. You have the ability to move beyond us managing symptoms of seizures and behavior struggles and towards a treatment that addresses the root cause of what is going on with our kiddos, and that's getting creatine to their brains, and it helps them get the energy they need to grow and learn. So when you think about your research, I want you to think about Eddie, a little boy who tries every single day to connect with the world around him, because what you're doing, it's not just science, it's his voice, it's his independence, and it's his future. As I end today, I want to tell you who Eddie is beyond his diagnosis of CTD. He's full of life. Everyone that meets him falls in love with him. All his therapists have told me they would kill for this little boy, which I'm like, don't do that, but they would do it. He loves to move around. He loves to explore, and he just loves life. He loves being a little boy, and he's taught us so much about just living life every single day to the fullest and enjoying the little things around you, and in his case, it's a stick. And he is capable of so much more, and with your help, we have hope that one day he can show this to the world. Thank you.
Next up, we will open with session 4, Cognitive Outcomes in Clinical Trials. Our first speaker in this session will be Audrey Thurm. Dr. Thurm is a clinical scientist and board-certified child clinical psychologist at Boston Children's Hospital. She's also a member of the faculty at Harvard Medical School. Her expertise is in understanding developmental trajectories of neurodevelopmental disorders, and this informs her work on outcome measures for clinical trials. She is the lead scientific collaborator on ACD's Parents 2.0 project, for which we are very, very grateful to her.
Good morning, everyone. I'm not going to have you do more exercise, but you're welcome to as we go. But I am here to present really, certainly on behalf of Heidi, who none of this would be happening without, but for all of you, because what we've done here, pretty much almost everyone in this room has participated in. And if you haven't, you're a new family. That's what we're doing is I'm going to now sort of show you the whirlwind of things. I'm just going to go through a few things that we've done. Really, it's the last 4 years, and this meeting and this summer, we're really capping off or finishing off an incredible amount of work that's been done. And I know later we're talking about clinical trial readiness. Well, this is the readiness for the clinical trial readiness that everyone's been able to engage in. So what is Parents Advancing Research Networks 2.0? It's parents, you all, advancing the work, and it's expanding on community tools and engagement. And that's the award that we've had for the last 2 years, which I'll talk about in a minute, which is a second phase of a first PCORI grant, and I'll explain what PCORI is and what the grant is. And it's called an engagement award because the Patient-Centered Outcomes Research Institute, which was formed less than 15 years ago, really is the entire focus of that institute is to empower, engage, and utilize communities and parents to be in the research, doing the research themselves, and to make all research parent-centered, patient-centered, and in this case, very much parent-centered. And so they have different types of awards, and one of them is for engagement. It's to help the families, the patients actually get engaged in the research. And the first project, spearheaded by Heidi, in 2021, while we didn't even have findings from a natural history study of CTD, GAMT was very early on in things. She had the foresight to think ahead about how to get families engaged, because none of it matters, none of the research matters if we don't have parents being educated, families being aware, involved, and creating their own research path for what they want for outcomes. And so the first grant focused on creating a core outcome set that is aligned with what matters most to families. So the model that was used really is this idea, and again, what CTD and the Association for Creatine Deficiencies has done is super helpful for this community. They also gave back to PCORI, and now many other rare disease conditions can use this model, and they have given it back to them to recruit an engagement team, which they've done, and there are parents in this room who've spent hours and hours and hours on this team to then recruit other patients to be part and parents to be part of a group that really formed. And it's why so many people are meeting here today, but so many people know each other well from hours and hours of Zooms across the world. And so parents trained other parents to be part of this community. They decided on the research topics, and in this case, it was the outcomes for studies. They built, and they have presentations, and they also taught each other how to present to groups and also to advocate to doctors. And then they shared all this, and we sort of then repeated for our next project that we've been doing in the last couple of years. So the first one, again, was creating what's called a core outcome set. And I just want to point out that CTD and GAMT are the first of rare disease conditions relating to neurodevelopmental disorders to actually publish a core outcome set. So cheers to all of you for doing that. It's really an amazing feat. And this is part of COMET, which is a worldwide effort, which again, it's mostly been, arthritis, knee problems, very common adult conditions that have done these core outcome sets. So we kind of, we're in the dark about how to do this for this kind of rare disease condition. And the idea is just that we, as a community, decide what's important. And there are many, many, many things that are important. We've heard about sleep and GI and so many things. But what is most important, so that as we launch the clinical trials that are now being launched, we can tell the companies and the researchers involved what is most important to families. And so again, there are many things involved, but the idea was, what are the most 8 to 10 sort of issues that are most important that would want to be treated in a trial. And it does exactly that. It allows us to make sure that different studies are collecting data on them, and that we can ensure that evidence builds on them. So this model is what happened over 2 years, from 2022 to 2024, in which the core outcome set was developed really by all the stakeholders involved, many, many parents, families, as well as other kinds of subject matter experts. So the idea was that it started with an evidence review of potential from the studies that were out there. Again, having done this before natural history was even published was a little difficult. And then many, many Delphi surveys that, again, many of you contributed to go over and say which is most important to me, and then a consensus workshop that happened 2 years ago. And so these are the outcomes that were decided on, and they were hopefully now what we're going to focus on a lot. The first is adaptive functioning, and specifically daily living skills that you've been hearing, all for the last few days, is super duper important. This goes from toileting to dressing to being able to feed yourself, be in the community, take care of yourself independently. Cognitive functioning, it goes without saying, and those of you who were at the workshop a few days ago heard that it is very much a priority, a little bit more difficult to measure, and we'll talk about that. Emotional dysregulation, we hear about behavioral outbursts, tantrums, regulating yourself. That's the sort of the term that is overarching. Not easy to define in a lot of ways in measures, because we know it changes. 2-year-olds shouldn't be expected to have the same control as 20-year-olds. Expressive communication, I don't need to say, what we just heard was a perfect example of that need. Fine motor functions, again, not to say gross motor is not important, but fine motor was just prioritized. And then MRS, brain creatine, seizures, and for GAMT, serum plasma guanylate acetate. So that's what was decided on. Again, that doesn't mean that other things are not important, but just as a core outcome set. So then we thought, okay, we're done. we know it's important. And then we realized, well, if there's a few more things to do, which is figure out actually how to measure these in trials and be able to provide some examples to researchers doing the work. And so for this, we decided that there's no way we could come up with actual measures, but what we wanted to do is come up with considerations, what's important to the families and how things are measured. And so we kind of grouped them together, is that the ones that have darker here are all what I would call neurodevelopmental outcomes. They're things that change with development no matter what. And so development is very important in them, and how we measure them depends on development. And the others are more, seizures is a little bit of an outlier, but the others would be considered biomarkers, or not necessarily the primary outcome unless we could figure that out. So how do we measure these things? The neurodevelopmental outcomes mostly require what we call clinical outcome assessments, things that are either on paper or performance-based, mostly that parents will fill out. Sometimes the kids will do themselves. Sometimes clinicians will assess. And then the others require either MRIs or blood specifically. So the idea for this second grant is that we wanted to continue to empower and engage parents and understand the qualities of measures that will both be acceptable to families and acceptable to regulators to be in trials, and have most importantly, have the possibility of measuring clinically meaningful change. And we'll talk about that a little bit later, but the most important thing that means is that it's meaningful to you all. We want it to be meaningful to the patients themselves, but in this case, it's the families who we're relying on to tell us, this is a change that makes sense and is meaningful and is different enough from before treatment that we're going to call it a success. And we know families do this every day when they're getting physical therapy, school, all the things they do, but it's a little different when we're thinking about a study where it would be a trial of a new treatment. And so the second grant, again, we sort of titled it Considerations for both CTD and GAMT in Outcome Measurement Tools. And the idea is that it would we would create something that would serve as a companion to the first, where meaningful changes in a clinical trial are recognized, and that tools would assess patient outcomes are sensitive enough to capture these changes. And that the idea is that we do not want a clinical trial to fail because of these measures. There's many reasons why a trial might fail, but we don't want it to be the measures. And so thanks to Emily, shout out to this beautiful timeline that was created. And we are here at the end of it. And so it's really been a feat, and we've done a lot that I'm going to go over a few of the examples of. But the idea is that we would engage parents in many different ways, have trainings, interview parents, have expert panel meetings, and I'm going to show you some examples of an exploratory research jam, which is a fancy different way of trying to have a focus group that is engaging parents in a different way. And then shout out to the caregiver community survey that's still ongoing that you can contribute to, and then highlight the alignment jam that we just had 2 days ago. So I'm going to go through some of the examples here of things that we've done. So these are the actual aims of the grant, which really was to identify measures used in similar types of trials, since we could not have any literature for CTD or GAMT trials so far. We had to rely on trials of other related conditions. And then we needed to identify caregivers' clinically meaningful differences, which we're still working on. But we used this research jam and a community-wide survey. And then we engaged a lot of subject matter experts to contribute to understanding what tools are out there, and comparing different tools, and then discussing it as we are right now. So the parents cohort is a group of parents, and again, shout out to Celeste right here, who really helped lead all this, that would, in this case, we had parents from the first round that could serve as mentors to new parents, and sometimes they were parents of very newly diagnosed individuals, children, and sometimes they were parents who had already been part of the first cohort, and that parents would teach each other and use their lived experience, as we're hearing throughout this meeting, to understand what's important and how to measure those things and provide feedback, which we had many, many Zooms where we were able to do that. And so again, thanks to Celeste for showing us how these powerful parents, starting with 4 mentors and then 19 cohort participants, were able to engage. And we were able to have a good distribution of CTD families, both male and female, as well as some GAMT families, and that was really helpful. And I just want to point out here that the fact that GAMT and CTD, which obviously are very biologically related, but still are different conditions, the fact that we did all of this across those 2 conditions is a model for how you can do this across conditions more broadly. And again, that's something that I think we are giving back to PCORI, and really, really important, because again, I'll say that there are about 1,000 of these rare disease conditions with outcomes related that need these models that these 2 conditions have been able to provide by grouping together. So this was the research jam that occurred, I think 1.5 years ago now, and the idea was that we would have parents participate, and we had 2 sessions for the time zones where smaller groups of parents could kind of participate and answer questions and think in different ways about measurement. the outcomes themselves, and then how we would measure them. So it included none of the experts, but an outside group of people that do this, that do jams as a living, and so it allowed for sort of different kinds of folks who don't know so much about this to ask the questions. And so there was 2 activities, and one was about core outcome domains, and participants did diverge on exactly which behaviors. For some, it was communication. For some, it was emotion regulation that were important to them. And we know that. That's why we don't have just one outcome, why there's many. But there was convergence on what would be appropriate changes that they would want to see. And again, for how to measure outcomes, that, here we're saying just what's the best way you could do it. There was some divergence on exactly which way. Different parents have different ideas based on age of child, their severity of symptoms, but there was convergence for some ways of measuring changes. And here's just an example of some of the findings that came up. For older kids, they thought, doctor checkups could be a good way, interviews, standardized evaluations, videos, just naturalistic videos. And one of the messages was not too much work for caregivers. Yeah, that's definitely something that was heard. But the overlapping themes were surveys could be helpful. If we can do home observations in a sort of objective way, we can do that. If we could use school evaluations, which has not been really tried so much in clinical trials, that was a message we heard. And then here are some examples for the younger cohort about how to do this. Now it's on us to say, how can we bring this to measures that can actually be validated, reliable, and have responsiveness enough to be used in clinical trials? And that's a bit of a tall task, so that's what we're working on. So then separately, we did this systematic review of literature, which again, could not include CTD or GAMT because we don't have clinical trials of these. But we did look at studies to evaluate the validity of outcome measures based on these concepts of interest, which were the outcomes, to explore how studies report on measures of these concepts. And so again, this is where we used just the developmental concepts. And what we did was a scoping review, even just the last 5 years, because there has really been an explosion of clinical trials, again, not for GAMT or CTD, but for many, many other neurodevelopmental conditions associated with intellectual disability, which cuts across a lot of these conditions. And so we, even just in the last 5 years, we were able to find, looking for these things, 79 studies of clinical trials published of related conditions that used these outcomes that we're looking at. And so what we did was look at which ones were used as primary. Again, the FDA requires that you put one outcome for the later phases as the primary. And so in these domains, and we found 25 that did use those. Let's see. And so we don't need to go into the details of what these are, but these are the ones that were found to be used the most. So for adaptive functioning, the Vineland, which measures many different domains. For cognitive functioning, the Bailey, which is a measure that allows for early developmental skills to be tested. For emotion regulation, the Aberrant Behavior Checklist, and so on. We found measures that have been used consistently, but we also found that a lot of measures were only used once in trials, and our main take-home, which the poster back there goes into, is that researchers do not do a good job of identifying which of these outcomes they're targeting specifically. And so it's really hard to go through this literature, and there's complexity in how we classify these things. So if parents are confused, it's because researchers are confusing them. We do not do a good job of these labels and defining in a lot of our studies. But again, we did find studies to go on, and we did find measures that have been used now consistently in several different trials that we can go on. It doesn't mean that it's what we will use, but we can start from there. Okay, so now I'm going to digress for a minute and talk about some of the other outcomes. So the plasma GAA, for those who were at the workshop, heard about this study that we were able to help initiate as us helping figure out what is the best method for doing plasma GAA, again, which we would certainly need in any GAMP trial. And so this trial is now ongoing, thanks to the work starting from the Curie grant, where we are now recruiting, and I think it's pretty well recruited, but we're still looking for some more, 10 patients to be able to see if we don't need to do full blood draws, but we can use finger pricks. And so the important idea here is that we're trying to figure out if GAA measurement is different based on time of day, medications, food. And so for these participants who will enroll, blood will be collected by fingerprint. I think it's several times a day for 2 weeks. And this is super duper important data now to show how we do it and when we do it best for clinical trials. So we're so grateful that this study is going on, thanks to Dr. Schultze, and we are hopeful that pretty soon we'll have findings from this, thanks to those GAMP families who participated. And then we heard yesterday about MRS, but I just wanted to bring it back that this is part of this grant, that for brain creatine, GAA, and phosphocreatine, we are very much realizing that just in the same way we need harmonization of our clinical outcome assessments for behavioral measures, we certainly need this for MRS. And I won't repeat everything that Dr. Cecil said yesterday, except to say that we need standardization and harmonization across any site doing an MRI that might be even diagnosing children with these conditions, because even the diagnostic, the first MRS would be important as a baseline. And so we would need that to look clinically and certainly in trials over time. So thank you very much to Dr. Cecil and the folks who worked on creating a paper which is now out for publication, and hopefully we will get this going. We're certainly going to need more funding to do this, but the idea is that we would have a straightforward way of using an FDA-cleared sequence for our MRS on all scanners that could be involved in trials and beyond, so that we can use this for diagnosis and tracking of GAA. I'll touch on seizures for a minute, because we focused less on seizures because we know people are tracking them for sure, but we do know that whatever way that seizures are tracked in a trial would need to be adhered to and be feasible and acceptable to families. And so we did do exploring. We heard from Dr. Kappel, who is a seizure expert neurologist, and she was able to give us some guidelines, starting with showing us that a video exists that's very, very helpful. Those at the workshop saw it, that can just explain what different types of seizures are, because it isn't always straightforward, especially when your child is young and you're not sure, as many of you know, what it is. Being able to use a video where you see observations of different types of seizures to help guide you is really important. And then different studies might use different methods, but these are the sort of very key components that would need to be captured if we're going to use some way of measuring seizure reduction. It might be length of seizure, frequency, all these different things, including whether emergency seizure medication is used as an outcome in trials. And then the last piece I want to focus on here is not which measure you use, but again, this idea of clinically meaningful change. And the field in general is just in its infancy with understanding this concept. And as you can see here, this is just a smattering of some of the different terminology that we've used. Some relates to statistics of what the measurement is, and some relates to just the idea of what is the smallest meaningful change that would come from parents that may or may not relate to actual units or scores on a measure. So our field is grappling with different terminology and trying to figure out statistically what's the best way to do this, but it turns out that no matter what we do, the input needs to come from the parents about what is clinically meaningful change. And so as part of this effort, we actually created a video to try to educate ourselves and then all of us in the community. And I think we'll try to put this out beyond, because we're really grappling with this. So if we can show this video, that would be great.
Trial. When clinical trials are done to evaluate treatments, they should focus on outcomes that matter to patients or their caregivers. Outcomes should represent something that patients or caregivers would like to see change in response to a treatment. Caregivers of children with different neurodevelopmental conditions may decide on areas that are important as outcomes for treatment. These outcomes can include areas like expressive communication, being able to express needs effectively; cognition, which is how you learn and think; daily living skills like bathing, toileting, and dressing; fine motor skills like manipulating objects in order to do things at home or at school; and emotional dysregulation, having difficulty managing different emotions. We now need to ask, what is a meaningful change in these areas? This is not only defined by how many points on a test their child gains to show improvement, but what skills or different behaviors would the caregiver want to see for them to say, this treatment is working? The changes a caregiver would need to see to decide that a treatment is working may be different for different treatments. Why? Treatments differ in how much time they take, how comfortable they are, and whether they have side effects or risks. How a caregiver defines a meaningful change for their child, or what they would need to see to decide if a treatment is working, might be different for easier, standard of care, and low-risk treatments than for treatments that are time-consuming, uncomfortable, or have more risks. This is because a caregiver might be thinking about whether a change in an outcome is worthwhile given what is involved for their child to receive the treatment or how much risk there is. We hope this video is helpful for thinking about questions like, what would you think you would need to see as an improvement for you to say, yes, this is a meaningful change? And is this different for different treatments?
All right. Well, I know that just probably raised more questions than answers, but I think that's the idea, is that we're still working on understanding this idea of not just meaningful, but worthwhile difference. And again, it depends on the treatment. It depends on how long you would be in the study. It depends on the age of the child. It depends on who you are and how impaired the child is, right? So these are things that we're working out, how to best ask these questions and how to then map these on to the measures that we have existing. And so I'm going to wrap it up now by saying, how are we putting this all together? Well, we definitely need all of your help in putting this together, because there'll be some documents that we'll certainly want to be vetting by groups of parents as we put this together. But this community survey, I think we had 55 complete at the beginning of the meeting. Maybe we have more now, but this is super, super helpful, because we asked these questions in the survey about measures, how long, how frequent you would take measures, but also questions about what is the outcome that's most meaningful to you, and what would be that measurement for you in skills that are developed that would say to you this is meaningful. And we simplify it by asking about low risk or high risk, but we need to think about exact studies and what that would mean. So we did all this. We did this alignment jam that we now have a lot to think about from. And now we're going to publish the systematic review that we put together of different measures from different trials. And then we're going to write a report about this entire project and hopefully put that out and publish things that are accessible on the ACD website and potentially beyond. And so that's where we are now. And I don't know if I'll take questions now, but I want to thank all of these people and Dr. Pasquale and so many others who have helped make the studies and things that we've done really come a long way so far in this effort. So thanks.
I just wondered if it was really clear on the community survey that that is part of the creatine info I am rare registry. So if you're wondering, why didn't I get the link or where's the QR? It is after you have consented and joined the registry, you'll see that as one of the surveys that you would complete.
So I just have a question about parent involvement moving forward, because I was a part of this second 2.0 and it was awesome. But I know that there are a lot of new families here that have joined in the last 6 months that are probably curious if they have an opportunity to take part in this as well. So is there going to be like a 3.0 or something for them?
That is a great question. Well, right now at this meeting, we're happy to just engage and talk for the next few hours for sure. Filling out that community survey will be super important. I believe that PCORI, again, I just want to say, PCORI grants are incredibly competitive to get. And so it was just such a feat for Heidi, who really spearheaded it, to not just get 1, but 2 engagement awards. I do think they cap you at 2 engagement awards. So we're probably done with that effort, but we will be thinking about different ways and certainly partnering with the industry partners that are working on these trials and trying to get more NIH funding to do more of this work. We're going to have to explore different ways to do this in the next stage. And I'll just say personally, I think, again, the fact that GAMPT and CTD aligned makes perfect sense, but potentially aligning with some other rare disease conditions, especially for resources to move this forward, I think will be important. But those new families, we absolutely want to hear from you. We will figure out ways.
I have a question. So I was, looking with interest on the slide that you had on the seizure, recording seizures and the guidance that is provided to caregivers on how they should be recording this. But there is a bridge, right? The way seizure is currently measured in clinical trials is extremely quantitative using an EEG, and the way we are proposing to record it for the purposes of observation and potentially eventual quantification is a different way. So how do we bridge that gap? I mean, the first mover who tries to do this in the context of a clinical trial faces the biggest risk. So what kind of, as an organization, as a community, what kind of pre-wiring can we do with the FDA or other avenues to make this more acceptable and lesser of a risk for those entities who might be open to using it in the context of a trial?
Yeah. Well, luckily, there is a very large set of developmental DEEs that where epilepsy has been the primary outcome for a long time, and many other conditions, Angelman's, that are in trials that have been documenting seizures, so we can rely on the ways they've measured it. I do believe that for most studies, actual overnight EEGs or EEGs themselves would not be outcomes, and it's difficult. And remember that even in this community where seizures are a massive issue, not everyone has them, and certainly to different degrees and different amounts of control from medications. So I think even though it will be certainly an outcome, it would probably just be one of these reporting ways of measuring it. And luckily, there are seizure diaries. There's even electronic, digital ways of capturing it that have now been used in other trials that I think we can lean on for evidence of their use. Okay.
Our next speaker will be Aurore Curie. Dr. Curie is a child neurologist at the Child Neurology Department of Lyon University Hospital. She's also affiliated with the Lyon Neuroscience Research Center. She has expertise in studying intellectual disabilities arising from rare causes. She has done an extensive amount of work in developing outcome measures for patients with intellectual disability, resulting in the Cognitoscope research platform. She will be speaking to us today about the CREAT criteria, work that she's been pursuing for the past several years in this space.
Thank you so much, and thank you for giving me the opportunity to present some of our results here. So yes, I'm going to talk about CREAT criteria study, a French study, sorry about that. So we did want to have an overview about the kind of outcome measures that could be used in clinical trials. And that's why, thanks to different fundings, extraordinary association, and also Ceres Brain Therapeutics, we were able, and also regular fundings, we were able to design this study. So, of course, you all know that it's very important to have objective, reliable, and sensitive outcome measures. And to be able to measure really what we would like to see, which means quantifies the core defects of the patients. So, of course, you know that CTD patients have major language delay, and and for boys at least, moderate to severe intellectual disability. We showed before that previously in a different study that 75% of CTD patients were not able to do a Wechsler scale, for example, to measure IQ. So here you can see the result in pink. It's the girls which are a little more adaptive abilities than compared to boys. So this study, we had a lot of objectives because we first did want to determine the most relevant outcome measure, and I think it's very complementary to the work that has been done with ACD, because we did want to try in a CTD population, a French one, but nevertheless. And we did want also to see if it was feasible to what we called single case experimental designs, which means to have the patient has its own control to be able to see if it works or not, but it means to be able to use outcome measures that can be repeated a number of times. We did want to see the feasibility of MRI study, and we did choose to be able to compare each patient to both a chronological age match control and to a mental age match control. So, in addition to that, we did want to perform an actual neuroimaging study, and I will show you some results from this too. We did want to have a cardiac assessment, and we did set up a bag collection at my hospital. So we included 24 CTD patients aged from 5 to 35, and each patient, as I said, was matched to a control matched in chronological age and to a control matched in mental age. In addition to that, we decided to include also girls. So we added 15 female, so 15 additional controls for each group. So 9 patients out of the 24 accepted to do a longitudinal study, which meant in our mind to have like 7 visits. 3 were on site and 4 were remoted. And we were able this way to test the outcome measure being performed at home. So we included a lot of clinical endpoints, So first, clinical endpoints, parental questionnaires, of course, cognitive, language, motor, social endpoints, a lot of different things. We did also add the cardiac assessment, and you can find the results in our first publication from this study. We did also include the biocollection and neuroimaging. I will tell you regarding the biocollection, a paper has been submitted, and the neuroimaging I will tell you more about. So 24 patients were actually included, all of them having a pathogenic SLC68 variant, and 54 were neomutations. The mean age, it was around 17 years, and the youngest had 6, was 6 until 27. So you can see here the age distribution. Regarding the girls, mean age was 19, coming from 7 to 36. Of course, now it's only preliminary result because we have only included 5 girls. So age at first symptom was roughly 12 months. Age at diagnosis for boys was 6 years. Not proud of this number, but it's the data. So the mean to diagnosis was through urinary test and spectroscopy for the most. As you can see here, a lot was on mothers from CTD patients because, at least in France, almost 80% of the mother has to decrease their professional activity to take care of their child. Regarding the female patients, you can see that the age at first sample was 9 months and age at diagnosis was even older because it was 10 years. So regarding the matching, you can see here that we matched it very closely. So the chronological age match controls were very similar to the CTD ones. And the same for mental age match controls, so 24 in each group. And you can see that the mental age was really very comparable. So regarding clinical endpoints, so we did a lot of family history, precise developmental trajectory, and we did want to have this kind of charts showing the different age at which each acquisition was made. So for example, here you can see, I don't know if you can see it, maybe here, no, there is no, I don't know, maybe this, yes. You can see here, so it's the months that are reported here, and in blue you have the CTD boys, in pink the girls, and in green the controls. So you can see the age at which each acquisition was made. So here again, for example, for walking, you can see that all boys were able to walk alone in our cohort, but you can see that they walked later compared to girls and compared to controls. And the same for everything, 5 motor skill, language. Here you can see that, for example, all girls were able to do everything before the age at which the boys were able to do it. But more than that, like you can, it was already mentioned, but like you can see only 40% of boys were able to produce a sentence. So again, the same with sociability. Here we can see that just only 50% of the boys were able to do games like pretend play. So we also we did want to have data regarding practical skills. For example, what age boys were able to get dressed alone. So 62% of CTD boys in our cohort was able to get dressed alone at 10 years. And you can see that for some it went up to 17 years to be able to do that. So we did also look at different things, taking a shower, everything we have already mentioned. We were interested also regarding reading, writing, numeracy, and we focused on behavioral issues because it's something that I think matters a lot. We also did want to have schooling trajectory. So here you can see, this time it's not exactly the same representation because it's a percentage. So maximum is 100%. And you can see that, for example, for girls, tantrums, my site is not so good. Difficulty with transition, it's also 100%. Motor stereotypies, social interaction difficulties, and interactions were very difficult for girls, and anxiety. So you can see that for some domains, girls were having much more behavioral problem compared to boys. Not saying that they do not have some, but. Then we performed also a very complete study regarding epilepsy, regarding like the description of the type of epilepsy they have, and we also did a centralized analysis of EEG. Then I cannot show everything, sorry. So regarding the data on physical examination, you can see the weight. It will not surprise you. They have the weight is lower than controls. We describe a lot of different things, such as hyperlaxity, And regarding balance assessment, you can see that in comparison to controls, CTD patients have difficulties with balance. It was shown with MEBC2 scale and SARA scales, which is a scale of ataxia. Regarding motor assessment, we also did want to have an idea on how the speed, the walking speed of the patient. So they performed a 6-minute walking test. And the good thing was that 100% of the patients were able to do that. As you can see here, the walking speed was lower for CTD patients compared to controls. We did also design, in our lab, we had 2 stairs, and so we measured the time it took them to climb the stairs. And as you can see, it was very interesting because it showed that it took them a much more longer time in comparison to chronological age match, but also to mental age match controls. So I will just mention the Purdue pegboard assessment. I think it's a very good tool. You can see here result for, I kept the colors are the same through the presentation. So in blue, you have CTD boys, in pink, CTD girls, and in green, controls. So you can see that Purdue pegboard was very interesting because it was feasible by 23 out of 24 patients, and it showed that they were really impaired, like the score was really lower in comparison to others. I will skip that. Okay, so we did also look at target symptoms defined with the parents because we thought, as it was already mentioned, but we saw that it was very important to be careful about what the parents thought was very important for their child. And we were able to do a visual analog scale and to see if it was repeatable, if it was stable when they repeated the assessment, and it was. So actimetry data were very interesting. We were a little bit afraid at first, but as you can see here, only 4 patients were unable to wear it. And it gave us a lot of data regarding sleep, quality of sleep, and also on activity during the day. And you can see here that it's difficult for them to stay still, and they are moving more than controls. So now, regarding the feasibility of performing an MRI without anesthesia, because in France, you know, regulation, maybe, I don't know, but it's difficult to convince them that it's really necessary to have general anesthesia for research. Maybe for a treatment, but not for an observational study. So, of course, it's a challenge to perform neuroimaging study because it's an unusual environment. They have to lie down. The antenna goes up to the head. It's a moving bed. It's difficult to understand. It makes noise. Even if you say that it makes noise, but it does not hurt, it's not easy for them to understand that. So, it's very important to stay still, and you know, as I do, how difficult it is for them to accept that. So, how was it possible for us to do that? First step, we used an illustrated document to see and to explain each step. So we first explained that they will have to lay down, and they can carry the paper with them, and of course use it through the assessment. So we used a mock scanner, and we did do it with gradual steps. So there were gradual steps in the mock scanner. They had to watch someone go inside. Sometimes it was not possible for them to accept that it would be a parent, but it was easier if it was someone from our staff. Then they had to touch the mock scanner and manipulate it, and it was a big win if they accepted to do so. Then to sit down on it, then to lay down on it, then to accept to have the headphones and the antenna over the head, and then to enter into the mock scanner. They practiced not to move, and I mean, the sound does not work, but it does not matter. It's just the sound of the MRI. So families were always welcome at all time, and I could not have done anything without the support of families from CTD patients, that's for sure. The kind of movie that we're using were not always Disney regular ones. For example, you can see the movie that was liked by a boy that went into the scanner. It was only train. This was the only thing that interested in. The second one, it's a family movie, but it was the only thing that could interest in the boy. So we were creative in this way. So we were able, and it was a surprise to us, we were able to do 17 out of 24 CTD patients, so 71% of them were able to perform an MRI without anesthesia, which we did not hope for at first, and parents and us were surprised. So we did 3D-T1, 3D-T2, FLAIR. We did also spectroscopy, long and short echo, basal ganglia and white matter, DTI and resting state. So here you can see some results. So here you have the, I don't know if you can see clearly, the peak, the creatine peak using long echo in a CTD male, and the same with the control. You can see that it's much higher. You can see here. Then you can see the same data at short echo, the peak, creatine peak here, and the creatine peak here. So yes, there is a difference. I added some data from girls because we have started to have also data on girls. And you can see that it's interesting. I don't know if you can see very clearly, but like the peak in girl is in between the very small peak in boy and the normal control peak here. And again, the same thing with short echo. So we did measure that using two different softwares. For the sake of time, I will just go direct to the LC model results. So you can see here the results with 17 CTD patients compared to 17 LC controls, so age match, as I mentioned before. We showed that there was a correlation, a significant correlation between the amount of creatine that we were able to measure using spectroscopy and the lighter scale giving us a nonverbal IQ. Then we did have this data, we described them, and we could compare, making side by side, CTD here and the chronological age match. And even if you are not a radiologist, you see that the corpus callosum here is thin compared to here, and they seem to have some kind of atrophia because the ventricles are larger than here. Again, another example. And here you can see, for example, the atrophy in the vermis of the cerebellum, and again, the larger ventricles. What we were able to do is that we use free surfer analysis to perform a volumetric and surfacic analysis. And we were able to show, here you can see, the volume of the corpus callosum. So in blue, you have the data from CTD patients, and in green, you have the data from controls. You can see that it is highly different. And here, we were really surprised because when we looked at cortical gray matter volume, we did not see any difference between both groups, whereas there was a strong decrease in white matter volume here compared to here. You see the size effect is really high. So now we are analyzing diffusion data to better understand the microstructure that might cause this in CTD. Regarding cognitive assessment, we did use lighter scale, and 23 out of 24 patients were able to perform this test. All girls were able to do it. So here you can see the results using nonverbal IQ, so roughly 40 in boys and around 60 in girls. But as you can see here, one girl had a normal IQ. Regarding progressive matrices, we did that also, and we showed that girls had higher mental age too. Regarding Vineland scale, we did show that the communication scale was domain was more impaired. Interestingly, they have better receptive than expressive skills, and a lot have a kind of strong point for domestic help at home. So we did use different questionnaires, MacArthur questionnaires for language, PDD-MRS for autism spectrum disorder, done sensory profile. I put you some results here. It allows us to know, what kind of specific visual or auditory or sensory processing the child has, and I think it's very important too. We did use also ABC, SRS, and it was important to us to look at the impact on primary caregiver. So we did use a CBI. We also use quality of life questionnaires. So here are the results, for example, from the ABC scale. I'm sorry, I have to, to keep moving, but everything is very interesting in my opinion. SRS2 showed that they had severe social difficulties, and it fits well with 75% of autism spectrum disorder patients within the cohort for boys, and all girls were autistic. So now I just would like to present the results from the 4 simple reasoning tasks on tablet that we designed specifically to be able to test CTD patients. So we did want to have very simple tasks with complicated verbal instruction. It had to be really very simple, and we did want to have a non-verbal assessment because of the problem with language in patients. So there are 4 levels of increasing difficulties, as you can see here. 96% of the patients were able to do the first 2 tasks, and 75% were able to do the third one. So it's very simple. They have to find the exact same picture as the one which is above. If you find the right one, it turns on it. So it's an immediate reward. We did use 2 different conditions, a matching condition where the neutral false response is different from the target, as you can see here, and the inhibition condition where you can have the feeling that it's a little bit more difficult because it looks like the target, but you should not select it. Okay, so it's the same with actual pictures, and I will go to the results. So we had plenty of data regarding controls, like it was validated on more than 150 healthy controls. So you can see the effect of age on reaction time. You can see here the results on error rate, and as you can see here, only children aged 3 to 4 were making a lot of mistakes after because it's very simple. So here are the results. So with 24 CTD, 24 age match controls, 24 mental age match control, and the 5 girls. As you can see here, CTD were slower compared to controls, but not different from mental age match controls, which was very important. They were slower than girls. Regarding the error rates this time, same population, same colors, you can see here that they made more mistakes than chronological age match controls, but also more mistakes than mental age match controls, and more mistakes than girls. So here you can see the impact of inhibition. It's important in CTD. A lot of them have a difficulty to inhibit some stimuli, and it's here they are making more mistakes for the condition when it looks like the target. Second level, the categorization. So now we had also 2 different conditions. The first one, when we had to put together 2 images, but you do not need to say that both of these are close. You just need to point to the answer. So it gives us the opportunity to assess things that cannot be assessed otherwise. So here you have the simple condition where the false response is totally different from the target, and the difficult condition where the false response is closer to the target, and it makes it more difficult to do. So here is the task on tablet. So you see it's going smoothly. Okay. So again, we had a lot of data regarding controls, more than 150. And again, what is interesting is that there is a decrease for reaction time. And here you can see that it's more difficult than the task before, as even older kids, like 9 to 10, they are still making mistakes. Okay, so same colors as before, but now for this task. So you can see that it's slightly more difficult because they are making more mistakes than before. But again, we showed that regarding reaction time, CTD patients were slower compared to chronological age match controls, but not different from mental age match controls, whereas girls were a little bit faster than boys. Again, here for error rate this time, you can see this strong effect. They are making more mistakes than chronological age match controls, but also more mistakes compared to mental age match controls and more mistakes compared to girls. I will skip this and go to the third task. So the third task was a visual analogical reasoning paradigm, and we got 3 different levels. The first level was very simple because all the elements were the same in the matrix, and here you have to select between those 2 responses. So this is what we call identical matrices. Then we had this one, which was slightly more difficult because you had to understand that the shape was changing here to find the right answer here. And here again, you had to combine the fact that both the shape and the size were changing to find the right answer. We also added to be inhibited false response, which means that, sorry about that, here that, it's more difficult because here the neutral false response, it's totally different from what is presented in the matrix. Whereas here, this you should not select, but it is present here, and it makes it more difficult for the child to do it. So the 5 conditions are here. So what we found very interesting was the fact that when we looked at error rate with this very short time, because this takes only a few minutes to do, it correlated very well with IQ level. Okay, we check also that it was feasible at home, and you can see here patients doing that at home, outside, and it did work well, and it was very stable over time, even when we looked at individuals. Regarding language assessment, we used PPVT5. So, for example, we showed 4 different images, and they had to show the picture corresponding to "show me to get dressed." Okay. So, interestingly, 23 out of 24 boys were able to do it with a lexical age of 5 years. And all girls were able with even better skills. Regarding expressive language, we use EVT3. What is nice is that we can compare PPVT, so receptive and expressive language, with these 2 tasks. So now they have to answer what is this, and of course it's a tree. And 17 out of 24 were able to do it, and all the girls. I just put here results from a different task assessing language. You can see in pink the results from the girls, and it's obvious that girls have better abilities regarding language compared to boys. But it's interesting to see that many tasks were possible for boys too. Now, just to finish, I will present the result of our 2 eye tracking tasks that we designed. So we did want to see, we projected 2 clips side by side. One was social, the other non-social. You can see different examples here. Please, the video. Okay. And so it's very simple because the child has just to accept to be in front of the screen and to look at the video. And you can see that afterwards we can design and we can compute the time they spend on the social region of interest and on the non-social region of interest. So we had also data regarding normal controls, and we of course got results for the CTD. So you can see here that they looked less longer compared to neurotypical kids. But nevertheless, what is interesting is that they looked more on social region of interest compared to non-social. even if you will see in a minute, it's stable over time. And the last task. So now it's not moving because it's not movie anymore, it's just pictures. And it's the same, like you have people that are interacting one with the other, and you have details in the room that are interesting but non-social. And then we can compute the time they spend on each. So you can see the redder it is, the more time they spend on the picture at this specific spot of the picture. And we did design that and for it to be acceptable by very young kids. And as you can see here, it's true that they are looking more at social than non-social, but not to the same extent as do mental age match controls. So, to conclude, I hope I'm okay. So, to conclude, we design new outcome measures to be able to targeting, really the ID dysfunction. And as you have seen in this task, we were very interested both in assessing reasoning, but also inhibition, because we think it's something very important for children. It is these outcome measures are appropriate to ID patients. I showed you results. They are validated in healthy children above 2 years of age and adults, and they can provide objective and quantitative assessment. The tablets, we showed that it was possible to carry the assessment at home. And the eye tracking, as I showed you, it's very interesting because it's very implicit instruction that just need to watch. So it's very simple. I hope that you were convinced by our neuroimaging study, and we hope that it could be useful biomarker in future study on CTD for each domain. I would not have done anything without the family, so I thank them really dearly here. It was amazing times for their visit to Lyon. And I also want, of course, to thank the funding agency that helped me, like Extraordinary Association, Cells Brain Therapeutics, and all the ones that contributed to the study, especially Marion Buchy, Farah Gerbi, and Pierre-Yves Postic, and Anne Chelus. Thanks a lot for your attention.
Thank you. That was really wonderful work. I was wondering about the MR scans you did without sedation. Looked like you did many sequences. Could you speak to the total duration of time that the scanning took, and also whether or not any of the subjects had a prior sedated scan, and if you could do a comparison on some quantitative measurements?
That's a good thing. Good question. Thank you. So I did put a hierarchy on the different sequences. So we always start with the one I really did want to have. And then if it's possible, then wonderful. But only if it's possible. So all of them, when I said that it was a success, all of them could have 3D-T1, MRS, T2 flare, and for the ones that could do DTI, we did DTI, and for the one that could continue, we did resting state data. The idea was during the whole sequence, except the last sequence, functional resting state, they could watch a movie, which was very important for them. For the one that used the train movie, the difficult thing was to take him out of the scanner because we had to stop the movie, and it was difficult. But that's all. So yeah, you're right. It would be wonderful to be able to compare. Unfortunately, most of the children had 1.5 Tesla MRI before, and I work at 3T because I think it's better. The signal to noise is better, but so I am interested, and we did compare, like clinically we reviewed and compare if there was a huge difference or not. But really, I think that we will have a machine difference that is not related to the child. But I am very eager, if anyone would like to help us financially, to continue this study because we would like to do, to do a sequential and to see after 4-5 years, if we repeat in the same setup the MRI, how is it going on?
That's great. And were you putting a head coil on for some of the sequences?
What do you mean?
Did they have an additional coil to the?
Yes.
Yeah. And
Yes.
did you have that for the whole time, or did you take them in and out? Okay.
I am very. This was possible because it was not done. It was done at a facility where we only do research, so it's not the same pressure. As at hospital, you have like 20 minutes and then move on. It's not possible with these kids, of course. But so we took the time. We use also attenuation signal device. So something that with repetitive sequence, it works well and it attenuates the level of noise. We did use, of course, a movie, and we also had a camera that they could not see but was on the mirror, on the antenna, and that allowed us to see if the child was moving or not. And if he was, I was communicating with him saying, you remember, you need really not to move. And it was very efficient because they could see that, and we could hear them because we had a mic allowing us also to hear them. So we saw them, we heard them. We did our best.
That's great. If there's no other questions, I have another question. How many regions did you take for the spectroscopy? How many voxel regions or?
2 different.
Okay, white and gray matter? Or?
Yes, gray matter and white matter.
Yeah, okay. That's great, thank you.
I don't have a question. Just as a parent with a female, thank you. That is the most data I have seen for the girls, and I appreciate you.
Thank you.
Hi, Aurore. I have a question with regards to the girls' study. I know it's not complete. You have 5 included, and you'll have 15. You said earlier that you have 1 girl with normal IQ. Is it relevant to include her? Does it distort the numbers, the results? Because if she has normal IQ, compared to boys, you have none with a normal IQ. What do you think about that?
Yes, it's a good question. Nevertheless, she has autism. And I think that for me, it's very important to include her in, because she is not asymptomatic. Huh? She has symptoms, but her reasoning skills are high. And I think it's important to acknowledge that in females, at least for what we have seen yet, the range, the spectrum of the abilities of the patients are really larger than in boys, I think. But yes, we did include her because we saw that it was a reflect of the female CTD population. So.